history of DNA. This is the DNA lecture, day 1. The first thing we're going to cover is a lot of the experiments that went on at this time when the scientists were trying to discover what DNA was, what it was made out of, how did it work, what did it really control. So the first one is going to be the Griffith. So Griffith experiment showed that live uncoated, or let's say it's shelled, it has a shell coating around it. So it's uncoated bacteria acquired the ability to make coats from dead coated bacteria. Okay, so he called the process transformation, which the study suggested that DNA was the genetic material. All that being said, this gets a little confusing. Here's an example or a diagram of what he did. He took a bacteria, which there were several different strands. This is a pneumonia bacteria. And he had the rough strand, which was non-deadly, and he shot up a mouse with that, and the mouse lived. So that's great. One is good. Then we have a different kind of bacteria, which is smooth and happens to be deadly. So when he shot the mouse up with that bacteria, the mouse ends up dying. So one's going to live, one's going to die. The next one, he takes a heat-killed, smooth, virulent or deadly strain, which is, if you noticed before, the smooth will kill the mouse, but this time he heat-killed it, so he killed all of the bacteria stuff that causes the mouse to die. So this one heat-killed and the mouse lives. So his next step was to actually combine a little sample of the rough, where the mouse is going to live, and also a sample of the heat smooth, where the mouse is supposed to live, combines those, shoots the mouse up, and the mouse ends up dying. Well, he had to figure out why did it die, and so he took a blood sample and figured out that the rough bacteria must have gained something from the heat-killed smooth, so when it replicated or duplicated itself, it ended up giving us a live, smooth, virulent, or deadly strand of bacteria. So something is being passed on between the two different bacterias. Now this is pretty much at Griffith's stopping point. He just figures out that something is being passed on. So if we go on to explain it, the smooth bacteria, the mouse will die. With the rough bacteria, the mouse will live. Okay, so he's taking the smooth bacteria, heat killing it, or getting rid of the stuff that is deadly, the mouse lives. Then he combines the heat-killed smooth and the rough, and the mouse dies again because something is passed on from the living bacteria and that smooth, the heat-killed smooth, to produce the living smooth, which is deadly. All right, so going on a little bit further, so we have the Griffith experiment that is showing something is passed on or transformed. Now, Oswald Avery is going to pose the question, which molecule caused the transformation of rough to smooth bacteria? Why did it change? Well, then he took this and he extracted that killed bacteria, the heat-killed bacteria, and mixed them with our 4 main macromolecules. Remember, we've covered lipids, proteins, carbohydrates, nucleic acids. All of those we've covered before. The lipids are fats, proteins are the workers of our cells, and carbohydrates, that's our main food sources. Nucleic acids, if you recall, is going to be DNA. So he exposed the rough cells to the macromolecules one at a time. Each time there was no transformation or nothing happened, nothing was passed on, except when he exposed the nucleic acid or the DNA. When he exposed that DNA to all this, his experiment, that's when something happened and it was changed. That's when we had the smooth and the heat killed that actually went on to produce the smooth, deadly bacteria. So he's figured out that whatever was transformed or being passed on has to be DNA. So Avery discovered that DNA is the nucleic acid that stores and transmits the genetic information from one generation to the next. And then we go on again to another experiment, and we have Hershey and Chase. In their experiment, they used radioactive labeling to identify DNA. They wanted to figure out what part of these molecules is actually the DNA, or the part being passed on. So they showed that DNA, not protein, is the genetic material of a bacteriophage, or a virus. Here's an example. Here's the picture. You're seeing the bacteriophage, which is the virus, and it has a protein coat, and it also has DNA. Well, they give it a radioactive coating so they can follow it, so they can see where it's going and what's happening, and identify it, so that they can see if it's going to be a protein or if it's the DNA that's being passed on. So a virus invades the bacteria. It consists of a DNA core and a protein coat. Let's go to the next slide. Well, it's a little messed up, but it's all right. All right, so we have our first virus, and you see it's a bright red. Well, that bright red shows that that part is radioactive. So it has a radioactive protein coat, or it has a radioactive shell made of protein. So he introduces the virus to that bacteria cell where it has DNA in there. And the product of that is just a regular bacteriophage, which is not radioactive. So in the process of making more bacteria and more cells, we find out that protein is not passed on, or else that shell, that bright red shell, would have been there. But we go on, and you see the red squiggle in the bottom, bacteriophage. That's showing that the radioactivity has been put onto that DNA. So the DNA is going to show how it is radioactive. The bacteriophage inserts that DNA, and it goes into the bacteria cell, and it comes out, and the DNA is still radioactive as the end result with that bacteriophage. So we now know that it's the DNA and not the protein that is actually being passed on from generation to generation of these cells. So DNA is our genetic information being passed down through the generations. Our next scientist is Erwin Chargaff. He figured out that we have 4 different bases in DNA. This is called Chargaff's rules. So we have equal amounts of adenine and thymine, and equal amounts of guanine and cytosine. So I don't have the ability right now to do this, but one of the things you need to remember is that A equals T and C equals G. No matter how much DNA we have, those things are going to be equal. A to T, C to G. And this really works, comes into effect a lot later when we start our copying, when we replicate, when we transcribe and translate. But you have to know A equals T and C equals G. So think of it this way. Apples grow on trees, A equals T, and cars go in garages. So it's C equals G. So Chargaff's rules, what he discovered, in every bit of DNA that he has worked with, A always equals T, C always equals G. Next up, we have Rosalind Franklin, and she is known for X-ray diffraction, or she worked with crystallography is what it's called. She took a large sample of DNA, and then she would shoot an X-ray at it. And when she shot the X-ray through the DNA, it caused a diffraction, or a breakup of that light, to show what DNA is really kind of looking like. And this is the first photographs, or the first images we actually have of DNA. So this image right here was taken in 1951. And the best way I can describe this X-ray diffraction is if you look at a chandelier and a beam of light will hit a piece of that glass. When it passes through, it breaks it up and you get a rainbow effect. Well, now the same thing can happen with this X-ray. The X-ray is shot through the DNA, and then it will break apart and cause our diffraction. Now, there are three things that it came up with showing, and these are really important. The first thing is that DNA has two strands. You can see because of the dark part at the top and the dark part at the bottom. So we have two strands. Second thing we figured out is that it is like a helix in its shape, which is kind of a great thing is it's a twisted ladder. So it's like a ribbon that's being twisted. And the third thing it figured out was that the nitrogen bases, that A, T, C, G that we got from Chargaff, show up in the middle of the image. So we have two strands. The bases are going towards the middle, and it is shaped like a helix or a twisted ladder, kind of like a ribbon. And those are three very, very important things that can lead to many, many exciting discoveries, which Watson and Crick took. So Watson and Crick are known for building the very first model of DNA by using Franklin's X-rays. At this point in time, it's kind of like a spy movie. We have several different sets of scientists that are trying to figure out what DNA is and how it looks. Well, really, Watson and Crick came up with model after model after model, and none of them seemed to work. Franklin came up with the double strand, the bases in the middle, the helix shape. Well, once Watson and Crick got a hold of one of her images, it wasn't but weeks later that they came up with the double helix model, which is what our DNA actually looks like. So they actually had to see her X-ray before they could come up with this model. Now, sadly, or whether or not, Wilkins, which was Franklin's partner, Watson and Crick went off to win the Nobel Peace Prize. Here's the sad part. Rosalind Franklin did not get any Nobel Peace Prize. She is known for coming up with X-ray diffraction, but she didn't get a whole lot of attention for it because she died before they could actually get the Nobel Peace Prize, and she died of ovarian cancer, which she probably got from all the radiation from X-rays. But anyway, that's a little bit of the history. So there's an image of Watson and Crick and their setup of the model of DNA. Now, what is DNA and how is it made up? We have the monomers of nucleic acids, and that's what DNA is. Deoxyribonucleic acid is a nucleotide. And these nucleotides consist of a phosphate group, a sugar, a 5-carbon sugar that is called deoxyribose. Deoxyribose is a sugar, so it's deoxyribonucleic acid, and a nitrogen base. Now, each of these, A, T, C, and G, remember, are the bases, will have this same general makeup: phosphate group, sugar, and a nitrogen base. So DNA is referred to as a twisted ladder. Here we are. If you see this right here, it does come up and it looks exactly like a ladder. Here's the rails on the side, and across this is actually the rungs. So, and then it is taken and it's twisted like that ribbon shape throughout. So the sugar and phosphate, remember each nucleotide has a sugar and a phosphate, is going to make up these rails on the side. So the rails are going to be sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way down. So they are alternating sugar and phosphate groups. So that is what's making up the sides of the DNA. And the rungs are going to be made up of our nitrogen bases. Sorry about the mix-up with the PowerPoint, but at this point I'm just going to continue. So these nitrogen bases are kind of like the steps or the rungs of the ladder. And they are joined across. A is adenine, T is thymine, C is cytosine, G is guanine. And they're joined across by hydrogen bonds. Now, hydrogen bonds, if you recall, are really weak bonds, which is a great thing that's going to be put into play when we go talking about DNA replication. So we have purines, which are a double ring structure, and we have pyrimidines, which have a single ring structure. And a purine will always pair with a pyrimidine. Now, what does that have to do with anything? Well, purines are going to be your adenine and your guanine, and the pyrimidines are going to be your thymine and cytosine. And one of the best ways to remember which is which is that pyrimidines has a Y in it, and thymine and cytosine also have Y's. So that is about one of the only ways I can kind of separate the two. Here is an image. You see your purines have the double rings or two circles, adenine and guanine. And then your pyrimidines are single ring structures, cytosine, thymine, uracil we'll talk about later. So DNA replication. Before a cell divides, DNA is copied during synthesis of interphase. Okay, going back to the cell cycle, you have interphase, which has G1, S, and G2. Well, S is synthesis, where we get DNA replication or making more DNA. During DNA replication, the DNA molecule separates into two strands. Each new strand will hook up with its complementary base partner. That means A is going to work with T, C is going to hook up with G. So A to T, C to G, always. And it will hook up with these base partners until it makes two new complementary strands or two identical strands. The strands follow Chargaff's rules on base pairing, A to T, C to G. Apples grow on trees, cars go in garages. The sites where separation and replication occur are called replication forks, and the replication is carried out by enzymes that unzip. Remember those hydrogen bonds in the middle of the ladder is going to be a weak bond. So we have this enzyme called helicase, and helicase is going to break the hydrogen bonds, in effect unzipping the DNA molecule. So we now have two separate strands instead of one double strand. The DNA polymerase are enzymes that create DNA molecules by assembling nucleotides, which, remember, the nucleotides are the building block or the monomer of nucleic acids. These enzymes are essential to DNA replication and usually work in pairs to create two identical strands from a single original DNA molecule. So, okay, here we go. We know from past lessons that ACE, if it ends in ACE, it is an enzyme. Okay, poly means many. So, where it says an ACE is an enzyme, it's going to start a reaction. That's going to start a process. Poly means many, so it's going to start the reaction, it's making many DNA. So the DNA polymerase simply makes more DNA. How does it do it? By popping in the A to the T and the C to the G. So we get the split by the helicase, and then we get the DNA polymerase to pop in the new nucleotides to make the more DNA. And here's an image that is happening. Here's our double-stranded DNA. In between them, on this left side, is a hydrogen bond. Well, helicase will come in and start unzipping it, the one in the middle. So it's widening out, separating, and now a DNA polymerase is going to start coming in and popping in the matching nucleotides. And it'll go all the way through that DNA molecule until we have two identical strands from that one strand. So DNA has been replicated where we have 2 from our 1. And then finally, let's talk about how that our DNA, which is pretty much 6 feet long, 6 feet of DNA in each and every one of our cells, and we have trillions of cells in our bodies. Well, how can all that fit into one cell? Well, the DNA coils around histones, and a histone is simply the purple ball. It is a protein. And then the DNA wraps around the protein, histone, and then those ball up together to make a bead-like structure, which is the many of those purple balls wrapped in DNA. That's the nucleosome. And then that's going to come in and coil together to make our chromatin. Well, remember, chromatin is how our DNA hangs out during interphase. And then when it goes to prophase and metaphase, our chromatin will coil up, tighten up, smash up to form our chromosomes, which are visible at prophase and metaphase. So our helix, our double helix on the far right side, wraps the histones, then makes the bead-like structure, the nucleosome. Then it starts coiling up, and it coils more until we are actually able to see it as the chromosome. So that's the DNA lecture of the history and the structure. I hope this helps, and we'll get to the next lesson next time. Thanks.